Hello, everyone. I'll do my introduction there then based on Ian's question in chat. It was very quiet. Everyone was indeed muted. We were just giving folks a few minutes to arrive and prep, but it seems like most people are here now, so I'll join in and say hi. So hello, I'm Sam Jones. I am the community scientist for the Cairngorms Rare Plants and Wild Connections project based up in the Cairngorms. I spotted Josie back in the chat there actually. So Josie works for the Cairngorms National Park, so it's nice to have her along. Yeah, so I done a little bit of waxcap work up in the Cairngorms, but I'm not really a waxcap specialist, so I'm going to be handing over to our experts today for the rest of the talk. I'm here to chair the session, to interact in chat, keep an eye on questions and things, and kind of manage things, make sure everything goes smoothly. But by way of a brief introduction, first of all, to Plant Life, you probably all have a vague idea who we are, but Plant Life conservation charity dedicated to conservation of wildflowers, plants and fungi across the UK and little bits internationally as well. And so, yeah, welcome to the Fall into Nature series. I should have checked which number this was in the series, but I know it's somewhere in the middle. There's been a few before it and some more afterwards. Go check out the others. I know they're all amazing. There's some really cool stuff going on, and I'm really excited for today's talk as well. Yeah, so welcome to our autumnal themed conversations. So yeah, keep an eye on everything because there's all sorts of interesting training opportunities and ways to keep in touch as well. But today we're going to be talking about waxcaps. The beautiful meadows fungi that come up at this time of year in all sorts of lovely colors. So we do, as I said, we do some work on waxcaps in the Cairngorms, but these panelists are going to talk in more detail about them. So first of all, Lucia will be coming on, who is Plant Life's conservation officer for the Magnificent Meadows Wales project, focuses on conservation of species-rich meadows. Meadows come with a magnificent array of fungi as well, and Lucia is an experienced fungal surveyor. And then we'll have Gareth Griffiths on later, who is from the Aberystwyth University, who's a fungal ecologist and taxonomist, whose research is focused on the ecology of grassland fungi, in particular the development of eDNA approaches to the assessment of grassland high fungal biodiversity. At the end of the session, we'll have questions, so we'll have a little Q&A session. If you could post those questions into the Q&A rather than the general chat, that would be amazing, because as you can already see with all the folks that are in chat, there's lots going on in there, and I'm not going to be able to keep up with all the questions that come in there. So we can use that for general conversation and discussion, and if questions could go into the questions and answers. I just spotted the questions already gone in there, so yeah. We are going to be recording the session for those who haven't already spotted, and Louise has noticed that, and the recordings will eventually go up on YouTube. We don't have a timescale for that. Hopefully it won't be too long, but check out the Plant Life YouTube channel for these recordings afterwards if you miss anything or if you want to show them to anyone else. But yeah, please put questions in the Q&A channel. But yeah, general chat for conversations and anything else you want to discuss. I'll be chatting in there as well with folks, and I'll put links in there, so keep an eye on that. We've automatically muted everyone, cameras are off, so no need to worry about that sort of thing. On the technical side, if you do have any technical problems though, just shout up in chat, whatever, and hopefully we'll be able to sort it out. Yeah, post links in chat. I think that's everything. Yeah, cool. Okie dokie. Well, without further ado then, I think that's us already. So why don't I hand over to Lucia, if that sounds good, for the first presentation. Always a bit of a fact when you're swapping over, give us a moment.
Right, so I think hopefully everyone can see it. Hi everyone, good afternoon. Thank you so much for joining us. Really great number, almost 200 people. Yeah, so my name is Lucia. I'm the conservation officer for a project called Magnificent Meadows Wales. We are funded by the Welsh government and work about meadows and grasslands, species-rich meadows and grasslands all around Wales. And some of the areas that are kind of orchid and flowerful in the summer can also turn into a really beautiful display of waxcaps in the autumn. So this is why Meadows project is also interested in waxcaps, because they are grassland fungi. And so what are waxcaps? I'll just jump straight in. So they are fungi in genus Hygrosidae. There are a few more now after the kind of latest genetics on them, but generally belong to this group. We've got 50 of these species in the UK, and they are mostly found in grasslands, as I said, which is quite I guess maybe unusual for fungi. People often associate fungi with woodlands, and it's true for lots of our kind of macro fungi, which with the beautiful big fruiting bodies, they do occur in woodlands. But for waxcaps, the opposite is truth. So most of them are found in grasslands, and we do have a few woodland specialists as well. And as their name suggests, they are characteristic for their waxy caps, sometimes almost slimy, and also really, really beautiful colors. They are quite small, so not huge kind of parasol size. They come from a few centimeters to maybe up to 15 when they are really big. And they can come in all the different colors of the rainbow. As you can see, just a small selection in my pictures, everything from white through reds to purples and blacks. So just before I start, I just wanted to kind of clarify a couple of phrases which you might have come across when you were looking into waxcaps, or you will probably hear us talking about in our presentations. So waxcap grassland, this is quite an interesting habitat because it's quite hard to actually put into one box. So when you compare it to our coniferous woodlands, for example, or our heathlands, we have quite concrete definitions of these, and we often know where exactly these habitats are around the country. With waxcap grassland, is not that easy because it can look like lots of different things. I'll show you some specific examples later on, but basically anything that's grassy and has got a big diversity of waxcaps is called waxcap grassland. The other term I wanted to mention is waxcap grassland assemblage. And this is basically like a joint name for our waxcaps, but also a few other groups of fungi that tend to prefer the same conditions and are therefore found in very similar places to waxcaps. So we tend to then kind of clump them together and call them waxcap grassland assemblage. So with waxcap grassland assemblage, apart from our waxcaps that we know of already, some other groups that belong to this assemblage are our earth tongues. We've got 12 species of these in the UK, and they are these strange looking black finger or earth tongue looking things popping out of the ground. Then we've got our corals, clubs, and spindles. And they can kind of look anything from more of the kind of individual points, like our white spindle at the bottom, to the kind of more branched and complex ones, like our beautiful violet coral, very, very rare fungus in the UK and globally. And yeah, it just looks like a coral. It's absolutely beautiful. And then we also have our pink gills. That's the last group that often gets clumped together in our grasslands with waxcaps, and they are named after the beautiful pink spores that they produce. So where do we find them? Before I kind of show you some very specific places and habitats, these are the kind of characteristics and requirements that they often need and are found in. So some areas that they really prefer, they need to be grassy. They really like being in undisturbed areas. And when I say undisturbed, I mean kind of mechanically undisturbed, which means no plowing allowed. This destroys the hyphae underground. Chemically untreated areas, again, they are super sensitive to any kind of chemicals. So any artificial fertilizers, insecticides, pesticides, fungicides is going to completely damage their underground root network called hyphae. And some papers say that they might take 50, 60 years to come back again. So no chemicals. They also prefer sites that are poor in nutrients. And when I say poor nutrients, that also can kind of translate into meaning that they are unfertilized. This can mean both chemical, but also natural fertilizers. And these three kind of characteristics of a grassland make a grassland is what we call unimproved grassland. That basically means that in the last 50, 60, 100 years, it hasn't been tampered with, it hasn't been plowed, hasn't been reseeded with anything else, hasn't been chemically treated. So this is what waxcaps really like, and often actually wildflowers as well in a similar way. So if you would like to encourage wildflowers and waxcaps, having soil poor in nutrients is really good. And the first step you can do to gain soil poor in nutrients is quite simple, is start raking the offcuts. Once you cut your grass or lawn, start raking the offcuts, and that's going to lower the nutrients. And they also quite like short sward, especially in the autumn when they are trying to pop up. They like well-drained soil and often are associated with moss. So some of the more specific areas where you can find them are a few different places, as I said. Meadows and pastures are really good. So meadows would be the places that have got lots of wildflowers, are generally cut for hay once a year by machines. Pastures, on the other hand, could have animals on them the whole year round. They are found in cemeteries, road verges. I'll come back to cemeteries in a bit. Also in grassy parts of other habitats like our heathlands and dunes and coastal areas. They can also be found in our amenity grasslands. For example, lawns and parks, especially lawns of stately homes, because these tend to be historically, again, unimproved. They might have been grazed for a very, very long time, but they maybe haven't been chemically treated. So they tend to be quite good for waxcaps. Cemeteries, I just wanted to leave to the last because I just love the fact that one of the best places to find waxcaps are cemeteries. And if you think about the kind of historical and land use point of view, cemeteries in our landscapes, they would have been there for hundreds and hundreds and hundreds of years sometimes, while everything else around them would be changed, would be built up on. Cemeteries from their kind of use point of view are there kind of almost protected in our urban areas. And because of this, the grassland that's there is also protected with them, which means that they wouldn't have been, changed into different crops, often not plowed or fertilized. It of course depends on how they're managed now as well. So if the cemeteries are still managed sympathetically, they haven't been treated, and also in cemeteries, they tend to keep the sward really short for tidiness. They also collect all the offcuts. So it's just all this combination of these requirements means that cemeteries are actually really, really great for them. So yeah, I think there is a good idea for your weekend walk. If you wanted to go and search for some waxcaps, go and visit some of your local cemeteries. So waxcaps are often, especially in ecology, in conservation, we can use them as indicator species. An indicator species concept is used across ecology. It's used from freshwater to all sorts of different habitats. An example for freshwater quality, for example, rather than looking at everything that lives in there, we can look at a specific group of animals to tell us something. So if we've got a big diversity of dragonfly larvae in our freshwater courses, we know that the water is clean. In a similar way, waxcap can also indicate something to us. And because of all of those elements that I mentioned, because of all of those kind of sensitivities that they are sensitive to chemicals and are found only in a really good grassland with a really good old soil. This means that if you find waxcaps, especially if you find a big diversity of them in one place, you are probably standing on a very good old grassland. So they can indicate to us that where they are found, the place where you are could potentially be an ancient or an unimproved grassland. Just from a conservation point of view, I just wanted to point out this, I guess, kind of an interesting fact about them, especially more recently. Waxcap grassland sites can be quite often overlooked, which means that they can be then lost to various different things. They can be then damaged by building on them. They can be overlooked, just left, not being managed at all, because they might not necessarily look like a really interesting site in the summer. Some places that have lots of wildflowers can also have waxcaps, but not all. They don't necessarily only occur in places that have big flower diversity. And this is the kind of problem for them, really, because if you go out and survey in the summer, which very often sites are surveyed in the summer, you might come across a site that looks like the one on the left. So if to an untrained eye, that can look like not very much. It's a pasture, you can't see very many wildflowers there in the summer. Therefore, you might just assume that there's not very much going on. This might be, you know, a site that will then get earmarked for new developments, for new tree planting schemes. Actually, quite an interesting conservation conflict there. But if you've seen, or if you haven't seen, we've got a webinar that we've done last year as Plant Life. It's called The Right Tree in the Right Place. Yeah, so but then come autumn, they can really change into these beautiful sites with huge diversity of waxcaps. So there's just one thing that we are kind of trying to get across as well, is that if you've got tree planting schemes and plan for trees, it's great, but really make sure that you are planting them in the right place, because this can damage some really good waxcap sites. So I just wanted to give you 5 common species that you might come across if you go out this autumn, and you might be able to identify a bit easier than some of the other ones. We've got our meadow waxcap, which is this kind of beautiful ivory color. They tend to grow quite big, and they can also sometimes be found in the circles. We've got our blackening waxcap, or also called witch's hat. And as the name suggests, it changes color slightly, and you can just see the tips of these waxcaps in the picture. They are starting to get black. We've got our scarlet waxcap. So this is one that's a bit more common than some of the other red species. We've got our pink ballerina, which you can't really mistake this for anything else. This is the only pink waxcap we've got. And the name, you can kind of see once the cap is a bit older, it opens up a bit, and it reminds us of a ballerina skirt. So this is our pink ballerina, beautiful pink. And our golden waxcap as well. So this is again within the yellow waxcaps. This is a common species and can be found in quite big numbers. But if you wanted to kind of get a bit more into identification of waxcaps, there are a few different things you can be looking out for. So color is the kind of obvious one that we already mentioned. Color will help you maybe you know, putting the waxcap into the correct color group, but then within the color groups, there might be quite a lot of species, especially some of the reds, yellows, and oranges. There's quite a few species within those colors. So once you've got the color group done, then you might need to look at some of these other characters to take you down into the species. So looking at the shape and the dimension, just the general size, shape of the cap is quite important. Is it conical? Is it flat? Is it pointed? Another character that's often used with other fungi as well as a identification character is gill attachment. This means also the spacing of the gills, but also the way they attach to the stem. Do they attach completely? Do they stop a bit before the stem starts? We've got texture, which is quite an interesting one. So waxcaps, most of them are more or less waxy, but they also can vary in their waxiness. So you might see in the fungi group, fungus books, terms for texture like silky, it might be flaky, it might be slimy, it might be sticky. So you're looking out for textures, both on the cap and on the stem. And smell. I really love this one. This is an interesting one. And the best way how to kind of concentrate the smell of a fungus is by either holding a piece of the cap in your hand and warming up, or putting a small piece in a Tupperware with a closed lid, a container with a closed lid, so you can concentrate the smell there. And this can sometimes really help you to tell the species apart. So within waxcaps, we've got species like cedar waxcap, for example, that smells like wood shavings. We've got oil waxcap, which smells apparently like fish oil. I haven't come across that one before. And we've got a honey waxcap, which really smells very strong, almost kind of like a manuka honey smell. But if you wanted to know details about the identification of different species, because we won't have time to cover it today, I'm just doing very generic introduction to waxcaps. If you're interested in a specific species and how to identify them, please look up some of our earlier webinars. We've done 2 different ones as a part of our project. They are both on Plant Life's YouTube channel, and I think some might be also putting some links in for you in the chat. So this one is really good. It's quite technical, but if you are interested in identification to species, it's really good. It tells you all of those different terms that you need to know, like the one that you can see umbilicate there, and all the wonderful words that you have in associations with fungi. And then also each species kind of broken down into all of these very detailed descriptions, so you know what to look out for and how to identify them. So just a couple of things to kind of bear in mind when you head out and you start looking for waxcaps, try to identify them. Just a couple of things. One of them is that they might change the color a bit with their age. 2 specific examples where it's quite obvious across the season are blackening waxcap, or also called witch's hat, and parrot waxcap. So with our blackening waxcap, they start off as really kind of pale, bright orange or yellows, and as you can see, as they get a bit older, they start turning black. So the middle picture you can see is getting a bit black, and the last picture there, beautiful photograph from our Facebook group, is the kind of older blackening waxcap, completely black. A good way to check this as well is even if you have younger specimens, is just to pinch it a bit, and it will start blackening after bruising. Another species that can get a bit confusing is parrot waxcap. So you can see on the very left, they start off as really kind of really rich green, sometimes quite hard to find in the grass, but they are very slimy, super slimy, these ones. But then they can change color as they age. And this is because actually with rain, the pigment literally washes out with rain. So they'll start losing the green pigment and will often start turning into kind of oranges, yellows. Sometimes, as you can see on the right, they can go kind of purpley as well. But one good tip to tell the parrot waxcaps is if you look at the very tip of the stem, if you turn it upside down, the tip of the stem should always have some green on it. Another thing to bear in mind when you are just starting your identification journey and looking for waxcaps is that they will change shape as they grow old. And again, this also means that trying to identify the younger specimens is a bit easier, because once you get to the older stages, it can be a bit harder because they change shape, they change color slightly. But you can see the change in the cap shape there from the younger ones that start as a very closed kind of conical shapes. You can see the pink ballerinas there, and on the left there we've got a crimson waxcap. And as they grow older, they first kind of open up into just flat discs. And then, as you can see the picture on the right of the pink ballerina, it also goes upwards and cracks. So yeah, there's just something to bear in mind. They're still the same species, but they can change the shape quite a lot. When to go and look for waxcaps? Very similarly to wildflowers, they will have their kind of ranges in fruiting, like with wildflowers, we've got early flowering species, late flowering species. With wildflowers, even if you go and search in the summer before the flowers are there, you kind of have an indication of what species it is from the leaves. Whereas with our waxcaps, we don't have that, so you kind of just need to catch them when they fruit, and it sometimes can be done and over with quite quickly. So we've got some species that start fruiting quite early in a season. Some of them might even start popping up in July, August, our citrine and fibrous waxcap. Then we've got some species that come a bit later on in the season, start in November and going all the way to January, for example, like our crimson waxcap. Then we've got some species that are kind of start early, but will keep popping up throughout the whole season until very late, like our witch's hat. There's plenty of them around now. And then we kind of have the rest, which do all sorts of different things. But generally, the peak season is now really is the perfect time to start going looking. October and November, between October and November, of course, it also depends on each year slightly, depending on the temperature, depending on the what happened in the summer, whether draft or yeah, it kind of slightly depends on the weather every season. But October, November is a really good core season where you can find the biggest amount of them in one place. So it's really kind of worth bearing that in mind as well. And if you've got a good spot for waxcaps, you might also want to visit multiple times a year because you'll have different species popping up at different times. Just to kind of finish off really on the resources, if you want any more information on the identification, there are a few really good things out there. We've got Waxcap ID leaflet, the plant life created, which can be found on our website, and you can download it for free. Then there are a couple of really good books, Grassland Fungi and the genus Hygrocybe B. If you just pop these titles into the Google, you'll be able to find them really easily. A guide to waxcaps in West Wales is really great. I love it. It's a smaller booklet, this one, which means that you can take it out into the field with you quite easily. It's got really good pictures. It can be found singly from the link that Sam will share with you, or you can buy packs of 20 from the Pembrokeshire Fungus Recording Network. So just find their Facebook group. That's the best way to get in touch with them. We also have our Facebook group that we started a few years ago. It's called Waxcap Watch. This was kind of done in conjunction with our citizen science app we launched a few years ago. And it's got now over 1,000 people, which is really amazing to see. And people share beautiful pictures of waxcaps there. We've got a few specialists that are quite active there. Will help you hopefully identify things to species. And I mentioned the webinars already. And just a special shout out to an interactive learning course called Introduction to Waxcap Grassland Fungi, Identification and Management, which has been created by our Scotland team. And yeah, it's a really amazing course. It takes you through all of the different chapters from ID to management to life cycles. It's got videos there. It's got kind of flipping charts and quizzes, and you can also test yourself after every chapter. So it's a really, really amazing resource. It should be available over the next couple of months, and the best way to find it once it's officially out, because it's just been kind of finalized now, is to head to the Scotland team pages, which again Sam is going to share the link with you. And I think that's it from me on the waxcap identification. Thank you very much.
Find the unmute button. There we go. Thank you very much, Lucia. That was really interesting. Lots and lots of good info there and a good introduction to waxcaps all round. Thanks, folks, for the few questions you've been putting in chat as well. We'll discuss some of them at the end. I know there's a few that I know Gareth's been having a look at one in text as well, but we might bring that up briefly for discussion. But yeah, so we'll lead on straight on to Gareth's chat now. So if you want to go ahead and put up your presentation. And I'll put that link in just as you were saying.
Okay, hopefully that's visible, is it? Yeah, brilliant. So first of all, thank you to Sam and Lucia for the opportunity to talk about waxcaps. And I'm going to follow on from what Lucia said and talk about a little bit of the studies we've done on sort of understanding their ecology, their rarity, and how to detect them, other than the standard fruit body surveys. So I'll move on. And the first thing I wanted to mention was the global rarity of these things. Quite often in the UK, we sort of make a lot of species that are nice to look at, and sort of a pleasure to see. I can quite often see, when I saw one earlier this morning outside the window here, a red kite. This is quite rare, or was quite rare in the UK. Now it's, due to lots of strident efforts by many conservationists, is now fairly common, but it was never globally threatened. And this is something I want to point out about the waxcaps. The waxcaps aren't just rare in the British Isles. In truth, they're more common in the British Isles than they are just about anywhere else. So we're a global biodiversity hotspot for these organisms. And obviously I'm going to be biased about this, but you don't have to believe me. You can see what the IUCN has to say, the International Union for the Conservation of Nature. It assesses the extinction risk for a wide range of species of plants, animals, fungi, and it assesses the citrine waxcap here as globally vulnerable. So that's the level of its extinction threat, which is the same level of extinction threat as faced by the snow leopard and the panda, which I think might surprise some of you. I should point out what they all have in common is you can't separate these things too much. It's habitat loss is the key, the main thing that's causing extinction of many species. And it's in the case of the waxcaps, it's the loss of ancient grasslands. I think Lucia sort of alluded to this idea of grasslands that haven't been tampered with, haven't been ploughed, fertilized, allowed to sort of proceed in succession to woodland. So that's what I mean by ancient grassland here. And the timescale I think she mentioned was about 50 to 100 years, which is quite a long time. You can get a grassland that botanically looks quite nice in a much shorter period than that. Oops. My slide isn't proceeding. Just to illustrate the point about the citrine waxcap. This is a GBIF image showing the global distribution of the species, and you can see that an awful lot of those dots are across the British Isles. So that's reinforcing my point about how the habitats we have, these ancient grasslands we have, being a global biodiversity hotspot. Not just for this, there's maybe a dozen other waxcap and other grassland fungal species that are similarly threatened. And there's a couple of species even that are endangered, which is even more rare than the panda, similar extinction risk to the blue whale. Anyway, so we have to do something about this. So I've got a 6-point plan, if you like, or what I'm going to try and cover briefly in the time I have. Awareness raising, some sort of legislative framework to protect these species. You also need to provide evidence of decline to show that things are getting worse. Some advice for people who own some land or manage land where these fungi exist. And then I suppose what I spent most of my time doing has been on the last two, trying to understand the ecological niche of these things. What role do they play in the grassland soil ecosystem? And does that help explain why they require certain particular conditions and are, negatively affected by others like fertilizer addition? And then also, I'll tell you something about how we're trying to assess the distribution of these organisms a bit more quickly, if you like. It's a bit high-tech method. It's not the most accessible method, but I'll tell you about the eDNA approach we've devised. So first of all, I have to awareness raising. Well, my awareness was raised by Maurice Rotherow 25 years ago. Maurice is sadly no longer with us, but I always like to mention him in any talk I give, that he raised the profile of waxcaps and grassland fungi generally across the UK. He was following the lead of this gentleman here in the red coat, Yves Arnolds, who's a Dutch mycologist, who was also very important in that. I guess in the Netherlands, it's a smaller country, it's even more intensively farmed, and I think Yves had estimated about 200 hectares of waxcap grassland remained. That's quite a perilous situation. David Bortman's contribution by producing this book here was important because that just spread the word. It meant we were all singing from the same song sheet. I should point out, I think it's been mentioned already by Lucia, that you have John Dunkelman's book on the grassland fungi. It's excellent. And also, I've got a picture of it later, which is this one from David Harris, Guide to Waxcaps of West Wales. I guess we could put a link about how to get both those in the chat at the end. They're both very accessible books and excellent. And I think both of them are on the call, so we should give them a clap for doing that. That's a really important contribution. From the scientific side, there's myself and a whole load of colleagues here. As you can see in this paper, I've been trying to work out the relationships between the different groups of waxcaps. So if you're annoyed about the names changing, it's us lot you can blame. So apologies for that. That's the way science works, I'm afraid. More broad awareness raising involves just getting citizen science, citizen input, where I think this webinar is very important for that. I think this is a plant life project from more than 20 years ago, the Pink Waxcap survey. And it was in 2000, and you can see the effect of that. The numbers of records, I mean, there's like 40 records across the UK in the 90s for the pink waxcap, and then it went skyrocketed in that year when Plant Life did their first initiative. But you can see also that how low it was before, that people weren't recording and recognizing a really obvious pink mushroom. You can see that here's awareness raising from Gary Easton amongst some school children with a rather dangerous looking hat. And here you've got John Bailey and Doreen Bailey in the West Country, who managed to get some quite good attention in the Times and the Daily Mail for their sort of discovery of wide range of waxcaps at Tinsfield Hall, I think it was. If you find these populations and they're threatened, it's important to be able to have sort of legal protection installed if needed. And this was an important court case back in 2005/6 in Llanishen Reservoir in Cardiff. So in the northern suburb of Cardiff, where Llanishen here, you can see the houses here. There was a reservoir that used to feed Cardiff, no longer used, and the owners, Western Power, wanted to drain the reservoir and build houses there. But unfortunately for them, it was discovered that there was a whole load of waxcaps along these grassy embankments here. It's only about 6 hectares of grassland, but there were 28 species there. So Countryside Council for Wales, as it was then, notified the site as an SSSI. Western Power didn't like that, so they undertook a judicial review, which they lost. So fortunately, the site maintained its SSSI status and has to be managed as such. It was helpful that you had sort of, this is a fairly wealthy suburb here. You have Julie Morgan, whose husband at the time was the First Minister. There were certain things that were helpful. I often reckon that had this decision gone the other way, I might not be here doing this work, and I would have found something else to go and do, because this was a fundamentally important thing for saying waxcaps are an important component of biodiversity. So now I'm going to take you back to the first experiment where people were trying to work out anything about waxcaps. This is park grass, which is the world's oldest field experiment, which was begun in the 1850s by a couple of guys in Hertfordshire, Gilbert and Laws. And it was mainly aimed to increase agricultural production. So they set up this field experiment here on a hay meadow, and all those little squares that you can see in the picture on the left are different fertilizer additions. One of the big questions they wanted to answer was how does nitrogen get from the air into the soil? So we all know now it's bacteria in the nodules on legume roots and free living bacteria that do that, but they didn't have a clue about that, and they thought it might be the fungi. The reason they thought that was because the fungi seemed to only occur on the sites, on the areas which didn't have any fertilizer added. So that just backs up what Lucia was saying earlier about the undisturbed grasslands. And you can see from the text on the right that they did find some waxcaps and Clavaria vermicularis, which is fragilis by now. So they recorded these things, and they also recorded there were only, well, the waxcaps were mostly on the plots where nothing was added. What they didn't see in that survey, and I'm pretty sure they would have noticed it and identified it had they seen it, was Hygrocybe punicea. But when I went there 130 years later, I found loads of this species there. So I can infer from that that in 1875, when they went there, the grassland wasn't ancient enough to host or to have fruit bodies of this species, whereas in the 130 years that went since then, they have matured and they're forming all these mushrooms. There was quite a lot. There were 71 fruit bodies on 2 fairly small plots, which are arrowed there. They're the only 2 plots on the whole site that receive no fertilizer. This wasn't a forever grassland. We do know that in the Napoleonic Wars, it was ploughed. So from about 1815, it was a hay meadow. So 60 years wasn't long enough for this particular species to colonize and reach maturity. So it kind of gives you an idea of the very long timescales these organisms operate at. Just to illustrate roughly the treatments, I just summarized it very simply. It's quite complicated. There's sites where they add nitrogen, some with no nitrogen but phosphorus, and then they have different levels of lime. And very quickly, you can see on this map the huge concentration of the wax caps on those plots that receive no lime or only very little lime and nothing else. And at the bottom here, I just got some diagrams, which I'll come back to later. They're to do with the eDNA studies. What we see as mushrooms is reflected in the eDNA in the soil, but I'll tell you about that later. I find this quite a compelling case for evidence of decline, because here's park grass. Here, this is London, and these are the surrounding vice counties, where and nature recording in the British Isles is done via these vice counties. You have to go about 50 kilometers from park grass to find the nearest other Hygrocybe punicea. The only thing about the control plots that makes them, I presume that they were an average piece of Hertfordshire, an average piece of Southeast England at the time. So it shows the level of habitat destruction that we've undertaken in the intervening period, that you have to go such a huge distance to find a fairly common fungus. So it shows that I think that's hugely compelling evidence of decline. Right, practical advice for conservationists. Lucia mentioned about how if you want to see your waxcaps fruiting, you need to keep the sward short in the autumn. So we did an experiment here to see exactly what was happening. The reason we did the experiment is because if you have to sort of to show the waxcaps, if you have to cut the grass short the whole summer season, then you have to choose, do you want waxcaps or do you want wildflowers and invertebrates? So we did various regimes here where we cut the sward to 3 centimeters from May onwards, or we delayed the first cut till late July, which is essentially a hay cut, but we made sure the sward was short later, and we had a super late hay cut, if you like. We didn't cut it till September, and then we cut it short, and then we had cuts here slightly longer, 8 centimeters all year, a hay cut that was 8 centimeters, and no cutting at all. So you can see that no cutting at all, as you'll often find, is gives you no wax caps. Not least, even if they were there, it'd be quite hard to find. But our more detailed investigations suggest they don't even make the mushrooms, and probably the reason they don't make the mushrooms is it wouldn't be very efficient for them in terms of the use of their energy and their resources to make a mushroom in the middle of long grass, because the spores wouldn't blow very far. But you can see, pleasingly, the greatest number of mushrooms was found on the hay cut plot. So that's great news, because it means that if you're managing a grassland habitat, you could just carry on doing your traditional hay cut. The only thing you need to be careful about is to try and make sure the aftermath grazing or mowing it leaves the sward quite short. And as Lucia did mention, I think, I'm pretty sure she did, it's really, really important to take the offcut away. Just pile it up somewhere. That will reduce the nutrient status as well, which is also helpful to promote these fungi. So we published that paper there. You can find it or email me if you want it. Right, now I was going to move on to the ecology of these fungi. They're really difficult things to work with, and you can't grow them. Most fungi, you can cut a piece of the cap or tissue and grow it on a petri dish in the lab. I've got one here. There you go. Don't know how well you can see that. You can see some plugs. You can see the plugs are furry. That's an Agaricus species, another grassland fungus. Waxcaps their spores barely germinate, and that's even after quite a lot of effort. So if we want to study their ecology, we've kind of got to do stuff in the field or do DNA-based analyses. So I was going to show you a few of those approaches that we've used. So the first clue about this is answering the question of what do they do in the soil? How do they get their food? Why are they sensitive to especially nitrogen fertilizers and so forth? So the first clue comes if you do a phylogenetic tree from that paper I mentioned about the phylogeny, the natural groupings of the members of the family Hygropheraceae, which is where all the waxcaps occur. And here you've got the waxcaps. Here's Punicea, Glyptiformis, Cytosina virginia. For a start, you can see that they're not all in one cluster. There are things in between them that aren't what we'd call waxcaps that live in grasslands. First off is Hygrophorus, some of you may know is a woodland fungus, and it's ectomycorrhizal, so it grows in association with the roots of trees. And the other one is some of them are lichenized. So it's lichen omphalia, which you may see around sort of in mountain tops around the British Isles, which is here. If you go to other parts of the world, you get proper lichens with mushrooms popping out of them, like Dictynema and Cora. So in amongst this family, you have some groups that are obviously what we call biotrophic. They have an association with some green plants, either algae or higher plants. So maybe that's offering us a clue as to why the waxcaps we get in our grasslands are quite difficult for me to grow in the lab, and it maybe explains their sensitivity to fertilizers. So one approach we could use, which involves taking a section of mushroom, is to do some isotope analysis. So you maybe hear about this in archaeological studies. They do this. They can have a look at the tooth of a 2,000-year-old skeleton, and they can work out where that person was born or where they did most of their growing up and stuff. So we could do something similar with waxcaps. I just illustrate the point with the marine food chain here, that you've got on the bottom, you've got the rarer isotope of carbon. This is not radioactive carbon. This is just a stable isotope of carbon. And then you've got the rarer isotope of nitrogen here. If anybody understands physics, it's got an extra neutron in the atomic nucleus. So it moves about a little bit slower because it's a little bit heavier. That's the basis of this method. I won't bore you with any more details. But you can see here that what happens as you progress up the food chain from the kelp and the algae and the primary producers at the bottom up to the polar bear, that the 13 carbon becomes more abundant. Those numbers there are in per mil, so 1/10%, and the nitrogen, the heavy nitrogen isotope, becomes more abundant as all the organisms lower down the food chain. The lighter nitrogen is lost in their urine. Anyway, so you've got an idea that you can have a look at the, if you like, the topography of a food chain using these isotopes. I'm sure there will be quite a few people who are vegetarian in the audience, and if you're a vegetarian, you'll probably be essentially, you're feeding lower down the food chain. You might be here, whereas if you're an ardent carnivore, you would be up here. You can also do strange things. You can, because corn and, as in maize and wheat, have different patterns because they're different physiologically. You can identify people who've been eating in the US versus people who've been eating in Europe because of different carbon-based things. So I've used those isotopic ratios to actually have a look at the waxcaps, and they turn out to be really strange. So here are the waxcaps, very enriched in that heavy nitrogen isotope and depleted in the heavier carbon isotope. If you take lots of ectomycorrhizal fungi, these are from British woodlands, they come out sort of less distinct. And if you take saprotrophs, they're down here. Saprotrophs will be things just feeding on dead organic matter, dead soil organic matter. And if you read many of the books, they'll tell you that waxcaps are saprotrophs. That's, I think, definitely incorrect. I think they are mycorrhizal, but I'm still compiling evidence to demonstrate that. I'll show you some of the evidence in the next slide. So they're doing something unusual. They're getting their nitrogen from a source in the soil that is already enriched. And if they're mycorrhizal, they're probably giving some of it, the lighter component, away to the plant, because that's the basis of a mycorrhizal relationship, is that the fungus gives nitrogen and sometimes phosphorus to the plant in exchange for sugar. So here's a bit more evidence about them being, whether they're mycorrhizal. So we have to consider two parts. We have the energy at the bottom and the nutrients at the top. So I showed you the isotope evidence for the nutrients. If you add lots of inorganic nitrogen, as you find with ectomycorrhizal species, that will kill them off. And that maybe explains why fertilizer shouldn't be added if you want to preserve your waxcap grasslands. A strange finding we did observe, and we're fairly clear about this, was that if you kill the soil animals, this wasn't us that did this, this was a bigger experiment with lots of biologists there, killing the soil animals led to the cessation of fruiting of waxcaps. So maybe they have some ability to obtain the nitrogen from dead soil animals, something strange like that. I don't know, I'm speculating there. In terms of energy, so we think they're mycorrhizal, and then you're going to ask me, what plants do they associate with? They seem to associate with a whole load of plants. Presumably in a grassland, there's loads of grass species, there's a high biomass of grass, but maybe they have particular associations with some of the various herb species. Maybe they're just a little bit Catholic in their taste of host. One last experiment we did was when we applied glyphosate to little circles where there previously had been mushrooms, killing all the grass. The following year, no waxcaps appeared in that circle, but they did appear around the circle. And we can see hyphae in here. That's got a little clamp connection there, so we can see hyphae inside the fine roots of host plants. We could detect the DNA. This is Hygrocybe conica that we did this on. So we found that. Some of my scientists are we're supposed to be skeptical and not just take evidence at face value. So I have colleagues who will ask me, additional questions to this. So I go and, I try and answer those questions, but the questions they're asking are becoming increasingly more difficult to answer. I guess that means I'm winning the battle, though, paradoxically. Right. So to finish, I was going to mention about how we detect waxcaps. If you, for example, I think in the question, was it Stephanie asked a question in the chat, and I thought I'll answer it now. It was about tree planting. So you have a grassland, say, for example, here. It's the springtime. Somebody's put the application in, they want to plant trees. They want to kind of do it that summer. So how do you answer the question, is this a valuable waxcap grassland? So here I present a method where you could do it. So what we do here is we've laid out a 30 by 30 quadrat here, and we'll take cores every 5 meters. So we have a bag of cores here, which we then freeze dry. That preserves the DNA and the biological component without them rotting. We freeze dry it, we grind it, and we extract DNA from a subsample of this. So it's less than a gram that we take out. So there's a big subsampling issue. We can't take all that soil. We can't extract DNA from all that soil either. So we have to subsample, so we have to mix things and be as careful as we can. The subsample we take is representative. Then we do something called PCR, which is like remember the PCR test for COVID, something akin to that. So we do PCR of all the fungal DNA in the sample that we've got, the DNA that we've got from the soil. And then we sequence a whole load of it, about 100,000 sequences for each sample of soil, which we can do with these, what they call next generation DNA sequences. These are the things that can do a human genome for like a couple of £100. So we're making use of medical advances and medical technological advances to do in the service of ecology. So what this tells us is what fungi are present, all the fungi, not just the waxcaps, all the fungi. And it also tells you the relative proportions of the different species. I'm going to jump this part, and I'm going to go to a case study that we quite liked, which was in Birmingham. This is the Leasows Country Park in suburban Birmingham. So it's a couple of miles from the M6. There used to be lots of factories here. It's only about 5 miles from where the whole global industrial revolution started. So it's kind of quite surprising to find a sort of little reservoir of wildlife here, mostly because this particular area of the park in suburban Hales Owen is quite steep. So that's probably why they didn't build houses on it. But there were a bunch of people here. This is Anthony, the wildlife ranger, and Perry. They look after the site. And this is I've forgotten his first name now. Nick Williams, who's here as well. And he was looking for waxcaps back in the 90s. Every year he'd go for walks and he'd find more and more waxcaps. And he found, by the time they contacted me, 22 different species there. And they were all quite keen to have this site labeled, notified as a site of special scientific interest. So what we did was we wandered around the best parts here, and in green you can see the areas where we took soil samples, like I showed in the earlier slide. And then we did the freeze drying, the extracting DNA for each of those samples separately. And we end up with a big species list. So each column here is 1 quadrat, and here are the different species. There were probably thousands of species on the original list, so I've just cut it down to only the grassland macrofungi, the earth tongues, the fairy clubs, the waxcaps, and the pink gills. And this is how we did it. One thing I haven't got time to sort of mention much of it. We find an awful lot of the fairy clubs, which weren't found by Nick and other people. That's probably because they're quite indistinct and small and easily missed. But we did find several waxcaps that they didn't find. We didn't find all the ones they had found previously, but we found some extra ones. So that means we could add to their species count, which is what they wanted us to do. So for example, we found Acuta conica, intermediate, and spadicea. This is very useful for them because Hygrocybius spadicea, the date-coloured waxcap, is it's a BAP species. In England, it's a section 41 species. In Wales, it's a section 7 species. So that's quite an important. It counts. It punches above its weight. It's like a trump card. So we managed to increase their species count to 28 because they'd not seen fruit bodies of 6 species. However, we did miss 4 species that they had found previously. That's because we didn't core all the soil. We're not going to find all the species unless we core all the soil. If you core all the soil, you don't have a site left. So what we did at the moment, eDNA, this is what we call eDNA is environmental DNA. This type of evidence it doesn't count for wildlife protection because the laws talk about mature individuals. So if we see DNA in the soil, we don't know if that was just a spore germling or lots of very small colonies. We can't say that colony was producing a mushroom. But what we can do is do something called eDNA guided surveying. So eDNA guided surveying is telling people exactly where a particular fungus was and guiding them to look very carefully. If you know what you're looking for, you can find it. If you've lost your keys in a grassland, which I've done more than once, if I know what the color of the fob was and I know roughly where I last saw it, I can find it more easily. So this is like, it's a directed search is much easier than a random search, isn't it? So we told Nick and Perry and Anthony, go and have a look particularly at these bits for extra species. And they found 4 species they hadn't found before. 3 of them, we predicted all these. In particular, this green earth tongue, Mycoglossum olivaceum, was very pleasing to find there. So the upshot of this was that that this site became notified as an SSSI 3 years ago, and I'm pretty sure it's the first site where eDNA evidence has been used to contribute to the legal protection of a wildlife site. And I just mentioned one other thing. There's other things that we can deploy this for. For example, here's the Welsh government use this method to see where if a farmer has illegally improved some land. If you want to plough a grassland that's considered sort of ancient, if you like, or long-lived, then you need to seek permission, and people from the government come out and do a screening just to make sure that you're not destroying a wildlife site. The same goes for tree planting as well. But here you have an example of a piece of heathland here where a big chunk of it was ploughed and was planted sown with wheat, and the farmer tried to deny that is what had happened, but the eDNA evidence demonstrated there were all sorts of fungi you wouldn't find in a wheat field here. There weren't actually any waxcaps, it was a whole load of other fungi that we used there. So this method has a lot of potential for wildlife conservation. And I need to mention these guides. David's on the call now, so he can answer the questions. I think he's on the call, so he can answer questions about how to get hold of these. And this is a picture of him and some friends doing some DNA barcoding. I mentioned eDNA meta barcoding, so I thought this is an appropriate point to just possibly deconfuse people. If you find an unusual mushroom, it may not be one that is in the book. It could be new to science. David found one last year that we think is new to science, and we're writing that work up. The way you could check that is to get DNA out of the mushroom sample, do the PCR on it, and then get a sequence for it. That's what DNA barcoding is, and that's amenable because you can do it with these things called bento labs. This is the type of thing you can do in your garage. You don't need a big expensive lab like we've got here at the university. It's a way that people can, if they're so minded, they can contribute very usefully to waxcap and fungal conservation. I'm pretty sure that this type of approach has been used for other groups of organisms, but it seems to be particularly useful as in fungal conservation. And I'd like to stop there and thank these many people here for funding and help over many years. So thank you for your attention, everybody.
Thank you.
Kept your time.
Thank you very much, Gareth. Oh, you're all right. Don't worry. It was very interesting. Thanks, Gareth. That was cool. Absolutely fascinating to mirror the words of Jill there. Gillian, I should say. That was excellent. Yeah, cool. So I think we'll move on to a bit of Q&A now. I know we've had lots of excellent questions posted. There's a few, I think, maybe quite speedy ones we could move through quite quick. So obviously not seeing fungi due to lack of rain. I think that's something we've all been experiencing this year, because I'm sure you folks will echo this as well. But indeed, we do. They do rely on the rain in this time of year. Kind of so often you'll see them delayed until you get the end of summer rains. Correct?
It varies from different regions as well.
Yeah.
So some people are saying they can see lots of waxcaps now, especially if they live in the uplands. There's people who are sort of in dry areas are complaining that they haven't seen anything yet.
Yeah. Cool. I'm trying to think if there's anything else we can cover quickly at the beginning. There was. Oh yeah, so a quick question about the hay cut. We can cover that one quickly. Lucia, you said you were happy to just explain what hay cut was, since we were talking about that in that. Gareth was talking about the end there.
Yeah, I was looking into this recently as well, because obviously working for a wildflower project, I was really looking into and making sure that we've got a way how we can manage for both wildflowers and waxcaps, and it's possible, which is great. So with the hay cut, the best thing is to do one cut, annual hay cut from Gareth's paper. The group that had the biggest amount of waxcaps was late July. I think I kind of looked into this, and I think there is a bit of a range, obviously, as long as it's quite short in September, late August. I think doing kind of late July, going into August, into mid-August is a good time for all things, which means that you can let your flowers to flower for a bit longer as well, which is really good for all of your insects, and then cutting it short come mid-late August for waxcaps to come. Also, if you obviously want to cover all of the increase biodiversity even more, always the best thing is to, maybe if you can, split your section into two as well, so you don't necessarily need to cut the whole meadow either, because again, that's not good for some of the insects and some of the flowers that might be late flowering things. So you can leave a small section uncut, and that way you can kind of make it work for everything.
Cool.
So I answer the question that Mariel's asked about wormers.
Yeah, sure. I was going to say it's kind of part of
That's
Aiden's question as well. So Aiden originally asked you the question about how sensitive waxcaps are to grazing as well. I know you posted that in the chat, Gareth, but if you can kind of cover both the grazing and the worming thing.
For the recording,
Yeah. Well, the grazing thing, people think that because grass goes in one end of the animal and poo comes out the other thing, and the poo is fertilizer. But if the grass that went in was from the site, then that's not nutrient enrichment. That's just moving the nutrients around the grassland. So that's a good thing, because it keeps the sward short. And also, if you have a pile of poo, I think the degradation and loss of nutrients from the habitat is more rapid. If you externally feed or additionally supplementary feed with silage, then that's less helpful. Anybody who's sort of got livestock animals would probably have to worm them. I think in some of these rewilding experiments, they're not wormed. And I know that the dung fungi are negatively affected by the wormers, because the wormers also kill fungi as well as invertebrates. But I don't think their effect goes into the soil as far as we know. There's no evidence that it does. It might do.
Cool.
Just working down the questions.
Yeah, thanks very much. Shall we go for the one from Stephanie? I know you kind of covered that already a bit, Gareth, but I'm not sure if Lucia wanted to
this is a hot
discuss
topic, isn't it?
the non-native forestry / native woodland creation stuff. If you're putting trees onto grasslands and meadows, how do you know you're not going to be putting on waxcaps if you can't necessarily see the waxcaps, so you're not surveying it?
I'm a big fan of extra tree planting, but I'm not a fan of where they want to plant them, because in Wales, it'll be the highest land, the marginal land is where they'll plant the trees. That land already has a high level of soil organic matter. So if you plant trees there, you're going to add very little soil organic matter, possibly because trees have the effect of drying out the soil. You might end up losing soil organic matter, which was the opposite of why the trees were planted.
Yeah.
So that's one thing. And also you would be destroying gradually as the trees grew, you'd be destroying the grassland habitat if it was rich in waxcaps. So what they should do is not plant on ancient grasslands anywhere, because there's plenty of non-ancient grasslands where lots of trees can be planted. The land that is no good for farming because it was ploughed, it was ploughed and it shouldn't have been ploughed. So that's where tree planting should happen. But at the moment, I think it's people like the EIA regulations are some of the regulations that are important for doing regulating tree planting. And I think there needs to be awareness raising among the people who do the screens that just looking for a diverse sward in the middle of summer doesn't tell you the whole story. You can have very diverse waxcap populations on what they call botanically mundane grasslands. The reason those grasslands have become botanically mundane is because they've been really heavily grazed in recent years by sheep. So that will sort of reduce species richness there. If you were to leave them, because the soil is good, the plant diversity would also come back quite quickly. Yeah, so I'm not really answering a question. I'm just agreeing there's a problem there, Stephanie. And if I said you could do eDNA analysis, we've done that a few times. NRW have asked us several times to do that. I guess the person who wants to plant the trees, the 300 quid or something it costs for an eDNA survey is small relative to the cost of planting all those trees, especially if it turns out they were wrongly planted and have to be pulled up 5 years later.
Or of course you can, if you don't have the access to the eDNA, you can also go and survey, but survey in the autumn for waxcaps, not in the summer or spring.
If you wait a bit, it's just I think these people who want to do planning stuff, they're usually impatient.
Telling people they have to go into two surveys at different times of year, I think can be a hard sell, can't it? Cool. Okay, shall we work down a little bit then? So there's a question about legislative framework. Can this seriously have an impact? I don't know what your opinions are, Gareth. What do you think about having an impact there? I suppose yes, with SSSIs protecting sites, but
it's taken a long time to get the staff of the statutory bodies who do the SSI notification to become aware of fungi. I think there's a good level of awareness now, but there wasn't when I started doing this. So they wouldn't consider fungi at all. It was a bit of a battle to persuade them about clinician, but when CCW went to war, they did it very effectively at the time. So that's how that battle was won. It'd be not EIA regulations. I think the people who do that are understaffed. That's the difficulty they have. So it's enforcing. The regulations are there. They just need to be enforced more efficiently.
Yeah, and I guess letting the councils also know about some of the sites. Like I know that for, I don't know if this is probably the case for lots of different counties. I was just looking at the paperwork. I think it was Hampshire Council and how they assess different sites for their local wildlife sites, which if you get the status of local wildlife site or a SINC, special area of natural, I never remember exactly what it is. Anyway, that kind of designation will mean that if there is a development coming up, these sites will be flagged up and will be more protected. And this specific document actually used the scoring system that Plant Life developed and can also be found in that first leaflet that I mentioned that can be found on our website. There is a specific scoring system that you can apply depending on how many waxcaps you find in the area. And if it's above a certain point, say 12, then this site should be considered as the local wildlife site. So if you know of areas like these, it's really good just to let your wildlife trust know, let your council know. Just any kind of extra protection they can get is good.
Yeah, absolutely.
As I can see that I think the Waxcap app that Plant Life had is still there, but I don't think it's been promoted this year, but it is still there. And you can basically geolocate species. Because as far as I can work out for biodiversity distributions these days, if it's not on a GIS layer, it's hard to account for it. So the more data that you guys in the audience here can ensure gets onto a GIS layer, showing particular areas where there are waxcaps, the better protection there'll be, because it'll be flagged up, and that will be maybe trigger a waxcap survey.
Yeah, and we actually had a really great success just very recently, kind of being confirmed from the app. I was working closely with Natural Resources Wales, and through the app last year, people were also taking pictures of the pink ballerinas. So we had a ground truth in proof, and these layers are now going to be officially included in the Natural Resources Wales habitat layers, specifically for tree planting requests. So if there is going to be a request for tree planting on a specific site that might have a pink ballerina, it's going to pop up. So yeah, hopefully the app will work again next year and we can map a few more sites.
Yeah, good.
John's asked a question about how far from a fruit body typical waxcap's DNA may be found.
Yeah, go on.
On the one hand, I don't know, but 99 point something% of the biomass of the mushroom is in the soil. Of the fungus is in the soil. So it's essentially, it's like viewing it like a flower on a big plant. You know, that's the mushroom. So I guess for different species, for Hygocybe punicea, that's quite a sort of a behemoth in the soil. It could be, you know, that could be a colony that was maybe 10 meters across. Maybe for some of the species that produce smaller mushrooms, there might just be a meter or two. But we know we know when we do that, even the 5 meter spacing of our cores, that occasionally we miss things.
Yeah, cool. I know if it's worth, I know you kind of already vaguely touched on taxonomy of waxcaps. Is it worth adding anything to what you had in that? I guess the impression I have of fungi taxonomy, because it's just a question about where do waxcaps fit into the fungi category. They're kind of all over, and it's very difficult to say with fungi, isn't it? Because how does it relate to what people understand of?
Yeah, when the phylum of the spore droppers, the basidiomycetes, the mushrooms.
Yeah.
So it's quite fairly closely related to things like the shop mushroom and I think pleurotus, the oyster mushrooms. That, you know, they've got the gilled fungi.
Yeah.
They're in that order Agaricales.
Cool.
One of the families. What's weird is the fairy clubs are very closely related to the waxcaps in that tree, and that's more surprising.
Yeah. So we kind of already touched a little bit about on being used for SSSI designation. The impression I have is no, not until very recently, and it's starting to become a thing. And again, environmental impact assessment, as far as I'm aware, it's not really part of environmental impact assessment standard, but we'd like to see it become more. Is that kind of the impression both of you have as well there?
Yeah, I mean, this is why I laboured the point a bit about these organisms being globally vulnerable, because that point needs to be reinforced. We've got international treaty obligations that none of the statutory bodies are particularly respecting at the moment, mostly because they don't know about them. So the more of you that tell people, you know, that the pink waxcap is comparably rare to the panda, and that's an important thing that people wouldn't know.
Yeah, cool. So Trisha posts, we have waxcap grassland assemblage. We have a waxcap grassland assemblage fungi in our churchyard. Will scarifying the surface area of the meadow in late summer, winter to sow yellow rattle seed damage the fungi? Scarifying's a funny one. I've heard it's kind of on the edge.
I'm really
is
glad
it okay? Is it not okay?
I'm actually really glad that someone asked this, because I think there are a couple of resources that keep mentioning the scarifying can be super damaging. And as far as we know, I hope that, this is what your experience, Gareth. Again, scarifying only happens once. You don't keep doing it every year either, and scarifying is only you are only doing a surface scratch, so it's not plowing. So as far as we know, it shouldn't be very damaging to waxcaps. Actually, there's a paper that I think Gareth was working on that showed that some scarification initially increased the numbers of waxcaps in an area.
I'm not sure if we did that. I was going to say with the yellow rattle, I think once you get, people are worrying a bit too much about that. If you sow the yellow rattle and you manage it correctly, it will be everywhere in a couple of years. It's not hard work for it to spread. So somebody's asked about foraging and eating these things.
And always the inevitable question. As far as I'm aware, none of them are edible, but I know
none of them are toxic. But then, you know, cardboard isn't toxic either.
Yeah, that's sort of level.
I think better to. I mean, I have fairly strong views about this, because it tends to go over the top about the foraging for fungi, that people just clear out everything in the whole area in the hope of, you know, eating just a couple of them. So it's a very wasteful and destructive activity.
Yeah, absolutely.
Something's mentioned about parasites of waxcaps. So all mushrooms are a very important home to lots of invertebrates. If you leave a mushroom long enough, you'll find them all generating lots of maggots. which might look themselves ugly, but their adult forms might be quite entrancing and possibly rare. So
Yeah.
just take a few for yourself, and just eat the tasty ones, the field mushrooms or something like that.
I suppose that's interesting to touch on. Is there other things that eat or parasitize or specifically feed on wax caps? I guess the fruiting bodies aren't a particularly significant part of their life cycle or productivity, though, so not anything specific. I don't know.
Yeah, we don't know. We know so little about what happens in the soil.
Yeah.
So there are all sorts of springtails that earn their crust by eating waxcap hyphae and this type of thing.
There's a question about, is planting a hedge around the grassland with wax caps just as damaging? I quite like that question. Yeah, that's an interesting one. I think obviously it would potentially be damaging in that very small strip where you will be disturbing the soil if there is lots of wax caps, but also at the same time you want to kind of cater for everything, and especially with conservation, you are trying to provide habitats for lots of different things. So, hedgerows have so many other benefits as an edge habitat, yeah, from plants to lots of other animals. So that's a very different thing to having a blanket tree planting.
Cool. Quick question just for the charm of it, because I kind of had this question too. Why are they colourful? What is the benefit to the wax caps of being colourful? You'd think there'd be a reason for it, wouldn't you? But I couldn't think of it before the presentation.
No, I've wondered that question and talked to lots of other biologists about this. I don't know, the main orangey yellowy colours, they're betylene pigments. So they're the same as in beetroot and the red campion. And the only fungi that have those pigments are wax caps and the fly agaric. It's not the red pigment, the fly agaric, it's the yellow pigment that's underneath that. It's really complicated. And the parrot wax cap pigment, nobody has the slightest clue what that is, because as soon as you try to extract it, it disappears. So, but we don't know why they're there. That's a question for the next generation of mycologists.
Very interesting. Cool. How best to increase population of wax caps on a grassland site? That's an interesting one.
Just keep on doing what you're doing.
Yeah, slow going. As Gareth mentioned, yeah, there are these numbers kind of flying around. It really depends on what has happened on a site before. So you really need to know the history of your site. If you know that it has been an arable field, it has been cultivated or fertilized, it might take 50, 60, 70 years for the soil to re-health and get all the interactions in a wax caps popping up. If it wasn't, yeah, just kind of keep doing the hay cut regime with removing your offcuts, or pasture with not huge numbers of animals with conservation grazing kind of guidelines, and it will slowly happen.
Yeah.
I just remembered you mentioned about scatterifying, and I couldn't remember. I know the experiment we did was to add ferrous chloride, which kills the moss.
Okay.
And that which is sort of a bit like we didn't remove the moss, we killed it, and that didn't have any effect. But that's because mosses don't have roots, so we think it's more likely that the plant partners of the wax caps are going to be higher plants, grasses or herbs, not mosses. So I think that's what it was. Not that I'm recommending adding moss killer to any site. I like the mosses.
Also, scarifying, we often actually recommend when you are seeding, doing it in strips. So again, they kind of, caters for lots of different things. So if you are worried about damaging a whole area, doing it in strips is actually quite a good way of, first of all, saving money, also saving time, because if you then sow it in strips, as you manage the site, the wildflowers, whatever you are seeding, is going to spread across. So yeah, that's a good technique.
Yeah, cool. I suppose the somewhat controversial question that often comes up in conservation circles. So why should we protect wax caps in general? I suppose aside from the moral answer of we should always protect all species if at all possible, are there any specific answers that could be given in the case of wax caps?
They may be the source of the next generation of antibiotics. If we kill them all off, we'll never know. I agree. If they were cuddlier and furrier, there would be more people on this webinar, I suspect. And equally, there'd be fewer people if we were talking about conserving bacteria. But they're all equally worthy of conservation.
Yeah.
And by saving some of the waxcap sites, you are very likely saving a very good, potentially ancient grassland. So there is quite a big overarching reason there.
Yeah. So if it's orchids you like, then orchids would tend to like ancient grasslands and might recolonize, even if if the grazing is modified to be more compatible for them.
Absolutely, yeah. And there's so little we understand about this, which has been highlighted intensely in this conversation. So we really don't know what we might be losing if we lose the waxcaps. Is very important as well to say, I suppose. Cool. I think we're more or less there now. I think there's one last little question we could say. Is there a size that a site should be, I suppose, for conservation purposes? I don't know. Is that too small for waxcaps? Is that a thing?
Oh, remarkable. You get little front gardens with. My mother-in-law used to live in a mining village in Scotland, and it was about 4 meters by 4, and there was a pink waxcap right in the middle of it.
I mean, in terms of kind of notifying areas and getting them protected, they often have actually the smallest size limit, which that can be then considered by councils to give it the protection, which I don't know the numbers now, it might be half a hectare or something. So for those purposes, there are some specific areas, but in terms of just conservation, can be anything.
Cool. I think we're more or less there then. So yeah, that's about us done. Thank you very much for joining us, folks, and thank you very much to our lovely panelists here for doing some excellent presentations and helping everyone with those really interesting questions. So thanks very much you for giving us such interesting questions. It's always great to have the conversation going on, the discussion and everything like that. It's so wonderful to have such interest in waxcap fungi as well. So thanks to Lucia and Gareth, and feel free everyone to go and check out the rest of the Fall into Nature series to see what else is going on out there. I think that's everything, unless either of you two had anything else you wanted to add to the finish off the talk.
No.
Cool. Cheers then, folks. Everyone take care and enjoy the rest of your week.
Thanks, bye.
Thank you, Sam.
Thank you.
You've got another one to do tomorrow, have you, Sam? Oops.